Hello again. We should continue with the central nervous system after finishing the peripheral nervous system. First, a very brief preliminary knowledge is to be given to you. Not much, but in a couple of weeks you are going to hear a detailed speech about the developmental main steps. So now what we need for today is the neural tube, which is gradually gets closed from the surface ectoderm, which surrounds the body. Anteriorly and posteriorly, the cranially, caudally, the process goes, and finally the tube becomes completely closed and becomes completely separated from the surface ectoderm. So this tube is needed for the build-up of the central nervous system, but there are other important structures belonging to the ectoderm. Outside the surface ectoderm, the neural tube is indicated here by this bluish color, but on the sides, both sides of the neural tube, there is a group of cells in a column arranged, and this becomes separated from both the surface ectoderm and the neural tube. That is the neural crest. The neural crest cells are needed for formation of the peripheral nervous system. Almost everything belonging to peripheral nervous system is the derivative of the neural crest. In addition to that, there is another piece of the surface ectoderm, just dorsal to the cranial part of the neural tube, and this one is called the placode ectoderm. Several structures develop, but we should not go into details. Now, what we need now is this neural tube. The neural tube at the beginning has got enlargements, 3 consecutive enlargements at the cranial end. The continuation of it remains quite narrow. These enlargements are the primary brain vesicles. The first one is the prosencephalon. Encephalos is the brain. Pro means standing in front or being in front. So prosencephalic brain vesicle, which is followed by the mesencephalic, meso in between, and as well as the rhombencephalic brain vesicle. This would need explanation, but when we get to the microscopy of the brain, you will understand why it is called rhombencephalon. The cavity of this brain vesicle has got a floor which has the shape of a rhombus. Therefore, it is rhombencephalon. So these are primary brain vesicles. And this bigger part at the cranial portion is needed for the brain. The brain is the most developed part of the central nervous system, so of course a bigger cell number is needed for making the brain. And continuation of the neural tube, which is relatively narrow, gives rise to the other part of the central nervous system, the spinal cord. With the rapid growth of these brain vesicles and all the neural tube, there are secondary enlargements of them, starting with the prosencephalon. On this ventrolateral side of the prosencephalic vesicle, other 2 vesicles are bulging out, and these become the paired telencephalon vesicles. The frame around the text reflects to the color on the drawing. So this is left telencephalic vesicle. You can imagine the other on the other side, and the midline piece remaining from prosencephalon is called the diencephalon. Dia is in between, so between the 2 telencephalic vesicles. Telos is the end, so that is the end brain. Between them is a diencephalon. The mesencephalon, what quite likely most will like, because it remains mesencephalon. There is no secondary derivatives, no secondary brain vesicles developing from it. The rhombencephalon, first a dorsal part develops from it. This one is the cerebellum. This is the final appearance of the cerebellum, just beneath the telencephalon. And the ventral portion is divided into a rostral and cranial and caudal part. The cranial is the pons, and the caudal is the medulla oblongata, the long brain, if you want. So these are the secondary vesicles. You can see how rapidly the telencephalon grows most, so very rapidly overlaps almost every other part of the brain. That is, during the phylogenesis, the fast-growing part of the brain. The final parts developing from these secondary brain vesicles are to be discussed briefly. The telencephalon consists of the two hemispheres, left and right hemispheres. Here you can see the right one, this mid-sagittal cut, so the left has been completely removed, and this is the extremely huge volume of the telencephalon on the right, so this is paired. Within the telencephalon there are connections, and these are the tracts. You remember the definition of the accumulation of nerve fibers, a bundle of nerve fibers in the peripheral nervous system that was called nerve. Exactly the same things within the central nervous system are called tracts. There are basically three types of these tracts. Commissural fibers are the ones which connect the two hemispheres, therefore necessarily they go through the midline. The biggest of them is this huge, very unique shaped collection of fibers crossing the midline. This is called corpus callosum. The other type is association fiber. Association fiber is the one which connects the points of the hemisphere or parts of the hemispheres on the same side. So they should not cross the midline on the same side. Almost every part of the brain is somehow connected to every other part of the brain, and that is the function of association fibers. And finally, the projection fibers. There are two types of these. One's carrying sensory information to the cortex up from subcortical areas, and these are ascending sensory fibers, projection fibers, and the opposite applies to the fibers leaving the telencephalon and projecting down to subcortical. So it could be in the spinal cord, for example, so anything below the cortex, if they end in, then we call them projection fibers. Then the basal ganglia, it's a very wrong word, because ganglia, you remember the definition, are accumulation of nerve cell bodies outside the cell, but these are inside the central nervous system, so rather nuclei would be the proper name for them, but we call them basal ganglia. So extremely important accumulations of nerve cell bodies are found in the telencephalic vesicle, as well as other small scattered in addition to the basal ganglia. The diencephalon, this one is midline portion, so necessarily at least the unpaired parts of the diencephalon have been cut through, but we start with the dorsalmost part of the diencephalon, which make the two thalami. So only a small connection, if it exists, is cut through in the mid-sagittal plane between the two thalami, interthalamic adhesion, not important the name, and below it lies the hypothalamus, quite easy to understand. Hypo is below, so this hypothalamus, which is midline structure, a single structure. There are smaller pieces attached to these main parts of the diencephalon. One is called epithalamus. Epi means above, so several small things, but amongst them, this is a very important part of endocrine system. This is the pineal body or pineal gland belonging to epithalamus, and again paired are the metathalami. Just behind the two thalami there are the metathalami, which contain extremely important relay centers for both acoustic and visual systems. The mesencephalon, I told you there are no secondary brain vesicles developing from it, but main portions are to be distinguished. Anything lying dorsal to the cavity passing through the mesencephalon, this one is the cerebral aqueduct. Later we should reach the topic again. So that is called the tectum. Tectum means lid. Ventral to it, to the cerebral aqueduct, the midline portion is called tegmentum, as well as continuing further ventrally. There are two very huge collections of fibers, and these are called the crura, the cerebral crura. Somehow these are the main branches, if you want, holding the telencephalon, but rather connecting the telencephalon with subcortical areas. So both ascending and descending tracts are making the cerebral crus. mostly descending fibers. So these are the divisions of the mesencephalic brain vesicle. The rhombencephalon, we mentioned cerebellum lying on the back, again undergoes quite a fast development. It consists of two hemispheres like the telencephalon did before. In between the two hemispheres, the midline connection is, because it resembles to a worm, therefore it is called worm, so this is the vermis. As well as scattered within the hemispheres and the vermis, there are nuclei, as well as there are peduncles through which the cerebellum gets connected with all the neighbors. So there are two superior peduncles connecting it with mesencephalon, two middles connecting with the pons, and two inferior peduncles connecting with the medulla oblongata. And again, these peduncles contain fibers going to the cerebellum or coming from, so afferent, efferent pathways of the cerebellum. These peduncles are made up of. The pons is just pons, medulla is medulla, but both of them face towards a cavity dorsally, and that is the fourth ventricle. Altogether, these three portions, the mesencephalon, pons, and the medulla, make up the lower brainstem. But widely speaking about brainstem, we should also include the diencephalon, which equals to upper brainstem, and altogether these are the widely speaking brainstem itself. These are to be known. Now, if neural tube gives rise to all these structures of the brain, the tube necessarily has got a cavity. Therefore, cavities of the primary and secondary brain vesicles remain in the adult brain, and these give rise to the ventricles, the ventricular system. Talking about these, there are the biggest cavities found with this. This is a drawing which shows only the ventricles plus shaded some main parts just to orient yourself. This one is corpus callosum, for example. We mentioned the biggest commissural system of the brain. Cerebellum has been cut through in the midline, so roughly you can orient yourself. So here was a big telencephalon itself, and the cavity within is the biggest, therefore, and these are the first and second ventricles. But which one is the first, which is the second, you should not care. These are the two lateral ventricles. Since the neural tube has got continuous lumen within, therefore the cavity of the telencephalon should have a connection with that of the diencephalon. This is interventricular foramen of Monro, through which they communicate. In the diencephalon, the cavity is called third ventricle, quite easy to understand if these were the first and second. The cavity of the mesencephalon remains quite narrow, so it does not enlarge as a ventricle. It is the already mentioned cerebral aqueduct belonging to mesencephalon. And to the rhombencephalon, the fourth ventricle belongs. Continuation of the fourth ventricle down to the spinal cord is the central canal, again considerably narrow, of course, a very small channel. As a very small channel, it extends all along the spinal cord. Now, all this system seems to be quite closed. There are only three holes through which the content of the ventricle can get out of the ventricular system. It is necessary to be done so because the content is cerebrospinal fluid, the fluid in which the brain floats, so necessarily should get out of the ventricular system towards the brain on the outer surface. Now, there are three holes on the fourth ventricle, one directed backward, and that one is called the foramen of Magendie. This is a single one, and there are two which are in the continuation of the fourth ventricle, extending in ventral direction, and at both ends of these continuations there are openings. These are the foramina of Luschka, so two of them exist, altogether three small spots, foramina, through which the content can flow out of the ventricular system. Importance of the ventricles is that not only containing this cerebrospinal fluid, but these are the sites, these are the sites or contain the structures producing the cerebrospinal fluid. And these are the very elaborate tiny systems of capillary loops, and these are the choroid plexuses. So we should go through the choroid plexuses of the ventricles, which part of the system does contain. So all these parts should be points or places for production of CSF, cerebrospinal fluid. Being the biggest, the lateral ventricle has got the biggest volume of this choroid plexus, so biggest volume of CSF it can produce. Not all the parts of the ventricle contain, but most of it contain choroid plexus. From here, the CSF can get into the third ventricle, on top of which there is also a small volume of choroid plexus, so additional CSF can be produced. Altogether, this flows through the mesencephalon, where there is no additional fluid produced. But in the fourth ventricle, again, quite a big choroid plexus is formed, so additional CSF is put to the previously produced one. And finally, all this can part of the very small portion of the CSF can flow down in the spinal cord, filling up the central canal. But most of the CSF flows out, so leaves the ventricular system and gets outside the brain. After ventricular system, very briefly, the blood supply of the brain is to be discussed. There are two pairs of arteries, one coming from the subclavian artery, and that is vertebral artery, going up in transverse foramina of the cervical vertebrae, except the last one, the seventh. Doesn't matter. The important is that through foramen magnum, I hope you are familiar with that, foramen magnum of the occipital bone, it gets to the posterior cranial fossa. This is one of the pairs. The other one is the internal carotid artery coming from the common carotid artery, and through the carotid canal it can get into the skull, into the middle cranial fossa, border between anterior and middle cranial fossa. The branches of them by which all the brain is supplied coming down, this is a convention when we talk about the general structure of the central nervous system. The talk is to be started from below, from the spinal cord, and gradually getting up and up. The reason for that is that the spinal cord has got less complex structure, so more easy to understand. So the same way the blood supply we used to describe. The two vertebral arteries getting into the posterior cranial fossa, giving branches, and these small branches are not needed, but quite easy to understand that the first branch should supply the posterior inferior surface of cerebellum. Doesn't matter. Important thing is that they soon, at the upper end of the medulla oblongata, they unite to form a single basilar artery, which also gives branches to cerebellum and to the pons as well as the last branch of it supplies the telencephalon. Therefore, this one is the posterior cerebral, and this is to be known by name. The internal carotid artery coming through the carotid canal gives a branch, a consecutive branch supplying the telencephalon, therefore middle cerebellar artery arises from it, as well as further continuing anteriorly the anterior cerebral artery. So these are only the main arteries to be known, supplying all the brain. Just for the sake of safety, these arteries have got anastomosis, so they are coupled with each other in a complete circle. So anything happening to any of the members of the group of arteries supplying the brain, the others can take over the place. This quite frequently happens. If you hold the brain in your hand, you can sometimes see a very narrow internal carotid on one side, and this one goes together with a much wider internal carotid on the other side. So the thing is that these are coupled into a complete circle, and this circle is called the circle of Willis. The venous drainage from the brain, it's enough to know that all the veins tend to dura sinuses, towards dura sinuses. When we get to the meninges, then I will explain to you what these dura sinuses are. Going down to in the central nervous system, the spinal cord comes. Tangentially, we mentioned in the previous lecture the general structure of the spinal cord. Now we should define it from where till where it lasts. So starts without any sharp border at foramen magnum as a continuation of the medulla oblongata, but it does not extend all along the vertebral column, but at L1, L2 it ends, usually ends, it tapers, becomes narrow, and finally ends at this level. L1, L2 depends in general on the body height. Of course, taller guys have got the termination at L1 and shorter ones at L2. It could be sexual difference, not considering me, but in general, male are taller and female are shorter. So males have got L1 ending and female in L2 ending spinal cord. All the spinal cord is made segmented, if you remember. The dorsal root ganglia have been mentioned before, sitting in intervertebral foramen. Not the same thickness the spinal cord has got all along, but there are two enlargements on it. One is on the cervical portion, or the other on the lumbosacral portion. Other enlargement is found here. If you remember the previous lecture, you remember that from this enlarged part of the spinal cord, two plexuses get origin. One is the cervical, other is the one which supplies all the upper limb. This one is the brachial plexus. And the same applies to the lumbosacral part of the spinal cord, from where the lumbar plexus supplying, as well as the sciatic plexus supplying the lower limb arise. So these are additional parts of the body, and therefore more neurons, more fibers are needed, and that is the reason for the widening of the spinal cord, for the enlargements found here. The segments, you remember, almost everywhere corresponding to the number of the proper vertebrae. The exception was here: 7 cervical vertebrae, 8 segments, 12, 5 again, and here was the other discrepancy where only 1 coccygeal segment existed, but that's what you well know now. Below L1, L2, only these roots or rootlets, also you remember the definition of them, originating from higher segments, these are found. So and altogether these resemble to a horse tail, and therefore it is called cauda equina. Equus is horse, so cauda equina is found below L1, L2 segment. You can see that because the discrepancy between the length of the vertebral column and the spinal cord, therefore these roots, dorsal ventral roots, should get more and more descending to find the proper emergency exit between the vertebrae, and therefore below L1 only these descending rootlets are filling up the vertebral column. This is just to remind you the importance of definition of the spinal segment, but hopefully remember. Still macroscopical structure, because these things are readily visible without microscope. Macroscopic structure of the spinal cord, briefly to be discussed. Hopefully you've heard about it in high school. Peripherally in the spinal cord, fiber tracts are found, and this makes up altogether the white matter, ascending and descending tracts. Now we should again go through the definition of the tract. The same structure found in the peripheral nervous system was called nerve, if you remember. The same in the central nervous system is called the tract. Posterior, lateral, and anterior funiculi, the fibers are arranged in. Between them, the borders are formed. Between the 2 posterior funiculi, a septum is found. Between the 2 anterior, there is a deep fissure. That is quite important to remember, since if you get the slide of the spinal cord, you must tell which one is ventral, which one is dorsal side, and if you find this very deep fissure, then that represents the ventral side. So anterior, and between the lateral funiculus and the two others, the entering dorsal rootlets, sensory fibers, make the division, and between the lateral and anterior, the exiting motor fibers mark the border. Centrally within the spinal cord, there are accumulation of pericardia. You remember the same accumulation of pericardia in the periphery was called ganglion. Here, these are nuclei. So all along the spinal cord, these are elongated nuclei, and these are called the horns. In cross-section at least, they resemble to horn, but if you consider all the length of the spinal cord, you can call them rather columns extending through. So the posterior, lateral, and anterior accumulation of pericardia are found at posterior, lateral, anterior horns, but the lateral horn is not everywhere existing one, but only there where autonomic pericardia are found, and it means that in cervical segments you can see no lateral horn at all. Posterior horn, because it is closer to the dorsal root, therefore it receives the sensory information. So in general, the mechanism is sensory, the function is sensory, and the opposite applies to the ventral horn, from pericardia, in which the fibers exit from spinal cord as motor fibers going to the periphery. The blood supply, briefly. That vertebral artery, what we just mentioned, related to the blood supply of the brain, as it goes up, it gives branches through intervertebral foramina towards the spinal cord, but finally, when it gets to the skull, it gives two posterior and two anterior spinal arteries, but the two anterior spinal arteries, very soon, you can see here, extremely small, sorry for that, but the two anterior spinals unite and only one extends down in front of the anterior median fissure, what I just mentioned before. Now, this supply of something like 30-40 centimeters long organ, the supply only from above, would be extremely unsafe and dangerous, and therefore there are several segmental reinforcing branches coming from different sources. Here you can see some of them originating from descending aorta. Out of them, one is usually very strong at the lower thoracic region, that is Adamkiewicz artery, but not important for now. But these are continuously joining the system, supplying the spinal cord itself, reinforcing all the system. The venous drainage is directed towards plexuses. First, there is one which surrounds the spinal cord within the vertebral column, that is internal vertebral venous plexus, from where the blood flows outside and joins another plexus around the vertebral bodies. The bodies are vertebrae, there is external vertebral venous plexus. The thing is that finally all the venous system joins the superior vena cava, mostly. Here is a drawing showing you how safely the blood supply is made. This one is anterior spinal artery, I mentioned before, the two posterior spinal arteries. This is a segmental branch flowing both dorsal and ventral roots to join either posterior or anterior spinal artery, and these are the anterior posterior and posterior posterior are connected with circular branches as well, so again making anastomosis between them makes the blood circulation more safe. I usually in a face-to-face lecture used to ask people what do they think, whether if you consider both these arteries, A and B, to be end arteries, it means that they have no connection with neighbors. So what do you think, which would produce bigger trouble? Closure of A or closure of B? It means closure of arteries supplying the white matter or closure of arteries supplying the gray matter. People used to answer B because the gray matter seems to be more important, but the proper answer is A, because the white matter consists of extremely long fiber tracts, and if on 10 micron thickness these tracts are cut through, then all the distal portion dies. So all the function of this tract is knocked out. Therefore, this is the more dangerous situation. Meninges. That applies to both parts of the central nervous system. Those are covered by 3 layers of meninges. Outside, so either lying inside the bony structure of the skull or the vertebral canal. So this is the outer layer, consisted of dense collagenous connective tissue. This is tough, therefore. So the dura mater is the outer layer, and there is a soft mother, which consists of 2 layers. One lines the inner aspect of dura mater, but it has got connections, very tiny, mini, delicate trabecules with the innermost layer of the soft mother. So the pachymeninges and the leptomeninges, not important names to be known. Soft and tough is enough. So the innermost layer of the soft mother is pia mater. There are general characteristics of these layers. So first, the dura is faithful to the inner aspect of either the skull or the vertebral column. So it goes wherever the bone goes and fuses with the periosteum of the bone. The arachnoid is faithful to the dura mater, so wherever the dura goes, arachnoid follows. Pia mater, opposite to these, is faithful to the surface of the brain. Doesn't matter what irregularity the brain produces, the pia has to follow this, has to cover the free surface of the central nervous system, either brain or spinal cord. There are spaces, even if some of them are virtual, so not really present, containing almost nothing, they are to be mentioned. One, outside the dura mater, the space, quite logically, should be called epidural space. And because the dura mater has got quite a good blood supply, any damage to these vessels result in bleeding, epidural bleeding, which, because the bone is not expandable, therefore the final result is the compression of the corresponding part of the central nervous system. So the epidural bleeding. In between the dura mater and the arachnoid, the space is called subdural space. This one is virtual, really contains nothing, but because the veins from the surface of the brain, spinal cord, should join veins formed by the dura mater. These are the dural sinuses. Therefore, they should necessarily go through the space, the subdural space, and if bleeding happens from them, that is called the subdural bleeding. Again, the final result is compression of the central nervous system. There is considerable, not virtual, but real, quite huge space between the 2 layers of soft mother, the arachnoid and the pia, and the name properly subarachnoid space, and this is the space in which the cerebrospinal fluid circulates. This is the fluid produced in the ventricles, ventricular system by choroid plexuses, and within this watery solution, the brain is floating. You understand that this is to reduce the weight of the brain, as well as giving some defense to it, but the primary is to reduce its own weight by floating in the water. Also, the vessels supplying the central nervous system or collecting venous blood from it are found here because they come outside or go outside the other layers of meninges. The dura mater of the brain, and here is the point where dural sinuses are to be briefly mentioned. So the thing is that at certain positions, the dura mater, the dense collagenous connective tissue, splits up and forms a channel. If this channel is lined by endothelium, then this is a beautiful vein. So the definition of dural sinuses, these are specific veins of the central nervous system, the wall of which is formed by the dense collagenous connective tissue. I hope you understand the need, the importance of this. A vein like femoral vein, what you've learned before, would not do well in a closed space where the brain is floating in a fluid. The weight of the brain produces pressure in the fluid, that is the intracranial pressure. And considering the general venous pressure in the body, hopefully you know that something like 60 or 90 100 mercury millimeters in the lower limb, reaching the abdominal region, it is something like 20 mercury millimeter. At the level of the heart is 0, above the heart is even negative. So the pressure in the vein would not be enough to keep it open. Therefore, the connective tissue wall is the one which keeps this vein open, letting the venous drainage to go. Several such dura sinuses exist, but maybe the biggest is to be known by you because you've learned it related to the skull, or the other big things like the superior sagittal sinus, or the continuation of it into the transverse sinuses, the sigmoid sinus. So you've learned the landmarks of these sinuses related to the skull, so quite likely you can imagine veins occupying these grooves of the skull. Other important characteristic of the dura of the brain, that somewhere, some places, it produces double layers. Here, for example, you can see a midline structure, mid-sagittal double layer of dura mater extending in between the two hemispheres, and this one resembles to sickle, and therefore the name is falx cerebri. Another thick layer of the dura mater, not horizontally but obliquely directed laterally. The lateral edge of it splits up into the transverse sinus and it gets united with the lower edge of the falx cerebri, and this makes a tent above the posterior cranial fossa where the cerebellum is located. Therefore, the name is tentorium cerebelli, tent of the cerebellum. The meninges of the spinal cord are in general the same. The only exception, what you can see, the dura mater here exists of, is made up of two laminae, two layers, external lamina, internal lamina. The external lamina is not the real dura mater. It is the one which fuses with the periosteum of the vertebrae, and because it lines the vertebral canal, therefore it is called endorachis. Rachis is the vertebral column itself. So the internal lamina is the real dura mater. The two layers of the soft mother are the same. So arachnoid attached to the inner aspect of dura mater, and pia, which is on the surface of the spinal cord, it consists of, and necessarily the spaces are the same. Between the two layers of the dura is the epidural space now, and within it there is the rich venous, internal vertebral venous plexus, a rich venous plexus. Between the internal lamina and the arachnoid, the virtual space is still called subdural space. And between the arachnoid and the pia mater, the space containing the CSF is the subarachnoid space. Because here the CSF is found, for diagnostic reasons, if CSF sample is to be got, then this puncture, the lumbar puncture, is used to be made. And because at L1, L2, the spinal cord ends, here you can see the very end of it, and only this cauda equina, the horse tail, is found here. Therefore, between L3, L4, L4, L5, a needle could be inserted without the risk of damaging the spinal cord. That is called lumbar puncture. Thank you for your attention, and I wish you good luck for the exams.